Welcome to this lecture. I am grateful to Professor Francesco Caminiti, President of the Italian Society of Physiology, who invited me to contribute to the physiology lecture program. At the same time, I am grateful to Professor Fiorenzo Conti, past President of the Society, for the idea to start the program. I intend to discuss a novel perspective in action control, which is rooted on the local nature of command function and on cortico-spinal modularity, rather than on the primacy of motor cortex in action control. Classical neurophysiology, in fact, suggests that motor cortex and its descending cortico-spinal tract play an exclusive role in action. This would consist in learning of independent finger movements, observing and dexterity, in the distributed encoding of movement parameters, such as force, velocity, direction, etc., and the adaptive motor behavior in the selection, gating, and gain control of spinal reflexes. This classical view goes hand in hand with the serial processing framework in which information flow through a unique unidirectional pipeline, first perception, then cognition, and finally action. However, this serial view is contradicted by a wealth of anatomical, physiological, and clinical data, and fails to account for the highly parallel architecture of the cerebral cortex. Considering M1 as the sole output of the cortex is an outdated view, which can be an obstacle for the interpretation of modern highly complex data and might even lead to a cul-de-sac if neuroscience continues to inspire machine learning design and network modeling. In contrast, there is evidence for different parieto-frontal spinal model that can bypass M1 and address motor command to the periphery to the spinal cord thanks to their private corticospinal system. These parieto-frontal spinal models observe different functions, such as arm reaching and its online control, eye-hand coordination, and tool use, conditional visual motor behavior, action plan switching and action suppression, spatial navigation and locomotion, mirror mechanisms, action initiation, motor sequences and time encoding, and defensive behavior. Now, the question concerns the evidence for corticospinal modularity. The first evidence for corticospinal modularity is the statistics of cortical connectivity. Cluster analysis allows defining cortical clusters, which are differentiated by colors here in the brain figurine of cortical and parietal areas, and in the consensus tree in both regions. Clusters are composed of strongly connected cortical areas that are spatially close, and they have strong local connectivity, as in small world network of graph theory. Clusters are the basic building block of models in our modularity hypothesis. In addition, frontal and parietal clusters are connected to each other in a selective fashion through long oligosynaptic pathways, and this is the motor dorsal premotor cluster, which is connected to a corresponding mediodorsal superior parietal cluster, and this is another connection which brings together information from the inferior premotor area to the inferior parietal areas and clusters. Fourth, which does not stem from cluster analysis but from the scrutiny of the literature, clusters often contain at least one area that projects to the spinal cord. This cluster projects to the spinal cord through M1, supplementary motor cortex, and dorsal premotor cortex. The ventral premotor areas issue two descending connection addressed to the spinal cord, as it does the cingulate motor area ventral, which is located in the ventral bank of the cingulate sulcus. Also, parietal cortex project to the spinal cord. Two projections arise from the inferior parietal cortex and one for the superior parietal cortex. The connectivity between parietal and frontal clusters shapes distributed parietofrontal modules containing central hubs, which are crucial for intermodular interactions. Good candidates for serving as hubs are those clusters that receive an equal number of projections from all other clusters instead from a few ones, and therefore show the maximum entropy. And these are the cingulate cluster, the motor dorsal premotor cluster, the posterior superior parietal cluster, and an inferior parietal one. This architecture would minimize communication distances, delays, and energy costs. The axon diameters, and therefore the conduction velocities and delays from parietal to frontal areas and vice versa, are variable and depend on both the area of origin and termination. In this slide, you see the projection that one superior parietal area addressed to motor cortex, dorsal premotor cortex, and cingulate motor area rostral contain axons with different diameters, which depend on where they project in the brain. Therefore, the conduction velocities of messages which arise from the superior parietal areas are different, as it is shown in this slide. Corticocortical connections are mostly made by small myelinated fibers, but also by thin unmyelinated one, and the separation between the two occurs at about 0.5 microns. And the last one, the unmyelinated one, conduct action potential in a variable and temporally dispersed fashion, requiring spatial and temporal summation to discharge their postsynaptic target. In contrast, the arrival time of faster myelinated axons varies by at most a few milliseconds. These corticocortical axons probably play a modulatory and/or a conditional driving role, as it is also suggested by the size of their synaptic button. The second evidence for corticospinal modularity is the multiplicity of corticospinal systems, each with its range of axon diameters and therefore of conduction velocities and delay. These descending systems are mostly composed by small and medium-sized myelinated axons, but also by very large axons. And they convey action potential at higher speed and with precise timing code, and in a more secure fashion as compared to the ipsilateral parietofrontal connection. Therefore, the existence of so many fast cortico-descending systems makes it improbable that all actions we perform are implemented thanks to an obligatory transfer of motor intention from parietal and/or from frontal area to motor cortex through ipsilateral, slower, and less secure corticocortical systems. The third evidence for corticospinal modularity is the timing of isomotor processing in the cerebral cortex. In this illustrative study, the onset time of activation of local field potential in different areas throughout the cerebral cortex and recorded in a simultaneous fashion during a go, no-go pattern discrimination task in which the animal had to categorize different visual stimuli and communicate the results of this categorization to the motor system by lever pressing, show that the earliest activation time relative to stimulus presentation in visual cortex occurs in a range between 50 and 70 milliseconds. But within this range, also motor cortex is activated, dorsal premotor cortex, frontal eye field, and also the earliest activation of far prefrontal areas follow within the late range of activation in visual cortex. The conclusion of this study showed that passing through the entire visual hierarchy to reach the motor output is not an obligatory step during visuomotor task in the paretofrontal system, since activation timing is mostly parallel across the entire region which has been studied. The fourth evidence for corticospinal modularity is the different consequence of M1 versus other motor lesion. In this upper panel, we can observe the nice precision grip posture that monkeys use to retrieve a small object from a hole. And this form of neck dexterity is allowed by the ability of monkeys to make independent finger movement, which are not anymore possible after lesion of motor cortex and of its descending monosynaptic corticomotor neural tract. In this occasion, given that the corticospinal tract is excitatory, the hand is also hypotonic. In these three panels, we have three cases respectively of optic ataxia, constructional apraxia, and ideomotor apraxia. Optic ataxia is consequence of superior parietal lesion. Patients show variable errors of reaching and then configuration. It is one of the pillars of the Balin syndrome, and it does occur not only in the lab or the clinical setup, but also in daily life. For instance, this patient, when trying to pour some water into a glass, they do it outside of the glass, and therefore is a rather debilitating disorder. As a very debilitating disorder is constructional apraxia, which consists in the difficulty of making a faithful copy of a model object. And constructional apraxia occurs mostly after lesion in the inferior parietal cortex and sometimes also of prefrontal cortex. Ideomotor apraxia instead consists in a difficulty of making an action under verbal instruction or under imitation. In this case, the patient was requested to make a military salute like this, and this is the outcome. This stand in contrast with the ability of this patient to make such actions as other one spontaneously in daily life. Optic ataxia, constructional apraxia, and ideomotor apraxia, which occurs after inferior parietal lesion and sometimes also after frontal lesion, are conditions in which patients are free of any sensory or motor deficits, and therefore these are a form of defective cognitive motor behavior rather than of motor behavior. The fifth evidence for corticospinal modularity is the metabolic cost of myelinated and unmyelinated axons, which here is shown across evolution. The metabolic cost of these fibers, when generating an action potential, can be calculated as the energetic cost of discharging the capacitance of an axon through its sodium channels, given that capacitance is a measure of the axon's ability to store electrical charges, and it plays a crucial role in the propagation on action potential across axons. Therefore, the energy required to discharge a membrane is proportional to its capacitance. Unmyelinated axons have small volume but high capacitance per unit surface area because they are bounded by only a single thickness or lipid bilayer, and they represent a compact solution to the problem of maintaining connectivity while contributing minimally to the growth in white matter volume, but they are metabolically very expensive, as compared to large myelinated axons, which have large volume but far less capacitance, and therefore for larger brain this cost decreases considerably because of the prevalence of myelinated axons. Therefore, let's now consider the process of myelinization across evolution. Across species from tree shrew to macaque, myelinization increased proportionally to brain size and brain weight, supporting the idea that longer conduction distances are associated with the adaptation of increased myelinization. Evolution predicts that in larger brain axons would increase in diameters to compensate for longer conduction distances, and the separation between myelinated and unmyelinated axons occurs at about 0.5 microns. Unmyelinated long-distance neocortical axons represent a case in which a premium is placed on volume minimization with the accompanying penalty of higher metabolic rate and slower speed, as said before. At the other size extreme, large myelinated axons are likely to play functional roles requiring speed or precise timing code, and these axons are only a small fraction of the total number present in the white matter and are a potential highly specific highways for neural activity. I will now discuss two typical cortico-spinal models among those which I introduced at the beginning of this talk, since they are illustrative of the parieto-frontal corticospinal modularity. The first model I will discuss is the hippocampal parietal cingulate-spinal model, which is formed by retrospinal area 31 and posteromedial parietal area PGM on one side, and the hippocampal formation and entorhinal cortex on the other side. And this model is supposed to perform a two-way transformation from allocentric to body center coordinates. At least this is the current conceptualization. The motor output of this model emerged from the cingulate motor area ventral, which is located in the ventral bank of the cingulate sulcus. The retrospinal area 31 in humans is activated during exposure to familiar place and by navigational tasks. And during spatial navigation, it showed theta power oscillation which reflect heading direction and changes, which is in keeping with what observed in rodents, where eight direction cells had been described in the retrospinal cortex as in the hippocampal region, which is connected to it. And these cells are target of CA1 field and subiculum, which are both involved in spatial navigation, as in the original O'Keefe and Ostrowski paper that started this entire field. In monkey, retrospinal neurons respond to large visual scenes, monitor eye movement and position, and this is believed to be a prerequisite to generate a map of the head in space. And therefore area 31 overall is considered as an intermediate node in the transformation from allocentric map in the hippocampus to egocentric one in parietal cortex. During locomotion, heading direction plays a crucial role, and the vestibular input is important for heading direction underlying spatial navigation and locomotion. This study from the Laquanitis lab shows the hubs of the vestibular connectome emerging from the analysis of data from the Human Connectome project about almost 1,000 LT human subjects. And on the medial side of the hemisphere shows that area 7A and 5L, which are the putative homologue of the monkey area PGM, plus several cingulate and retrosplenial area show a high degree of hardness. Area PGM belongs to the same cluster of retrosplenial area 31, and it contains neurons which are related to virtual active navigation, and their activity is route selective. The animal was trained to navigate in a special map following route number 4, passing through a number of checkpoint locations until he reached his final destination room, which was shown to him before at the onset of the trial. And you can see that neuroactivity varies depending on when the animal, where it is located, at which checkpoint, and increase significantly at checkpoint number 4. The path, which I will try to show you in a movie, is the following one, and this is the onset of the trial with the presentation of the destination room. The monkey is waiting in a delay time until the route number 4 is shown. He starts navigating by using the joystick. Now is at checkpoint number 4. He makes a left turn and it goes towards checkpoint number 6. It is at checkpoint number 6, and this is the destination room, and therefore it is rewarded since it made a correct trial. Area PG is located in the inferior parietal lobule and with PGM share cytoarchitectonics. And the neuroactivity in this area seems to reflect the mental abstract representation of very complex spatial maps, such as maze. And in this very ingenious experiment performed in Georgopoulos' lab, monkeys were trained to determine in a covert fashion and from a single fixation point whether a critical path through the maze reached an exit or a blind ending. This was an exit maze with a direct path, while this was also an exit maze, but it required a left turn at 90 degrees to reach an exit. These were no exit maze, which were shown to the monkey, of course. And during this process, neural activity was specially tuned to the maze path direction, and the population vector pointed toward the exit of the maze during fixation, despite no movement being made and no change in visual input occurred. Concerning the hippocampal formation, in this elegant study, neuroactivity was recorded by telemetry while a monkey performed a foraging task in an open space. And the neuroactivity was recorded from the hippocampal formation, from the entorhinal cortex, and from the subiculum, And you can see that the main results is that head height, speed, head direction, and tilt were dominating the activity which was recorded, together with eye movement. And the hippocampal that activity was intermittent since it was modulated by eye movement, the phasal location and 3D orientations, but not place dominated for spatial selectivity. Together with studies done many years ago, which had shown that cells in the hippocampus responds to visual stimuli and are sensitive to the direction of gaze, the conclusion is that the influence of eye and head variables might enrich the hippocampal activity, facilitating the active sensitive to guide navigation and locomotion by using landmark location. And the hippocampal activity combines different signals, serve as a multiplexer and might bridge the dichotomy between the representation of allocentric map in the hippocampus and egocentric one in posterior parietal cortex and in retrospinal cortex. Concerning spatial navigation disorders, in monkeys, muscimol inactivation of area PGM determines route-specific navigation disorder, as you can imagine from the movie that I showed to you a few slides ago. The hippocampal lesions seem to affect allocentric navigation rather than egocentric one. In humans, navigation tasks are impaired after retrospinal cortex lesion, as spatial memory is affected by hippocampal lesion. And in rats, lesion of the retrospinal cortex impairs a number of functions related to spatial memory, allocentric working memory, and egocentric memory. The motor output of this model can reach the motor periphery through the projection stemming from the cingulate motor area ventral, which is addressed to the intermediate zone of the spinal cord. And this region receive projection from both area 31 and PGM. The cingulate motor area ventral contain a visually related zone with sensitivity to the optic flow and vestibular information, which suggests a role in the control of navigation. And the electrical stimulation of this region evoke hind limb movement at a relatively low threshold, which is in keeping with the strong projection that it addressed to the spinal cord. And the role of this model in spatial navigation is supported by the observation that the motor cortex modulation on the rhythmic activity of the spinal locomotor centers is mostly associated to willful decision to initiate or to modify locomotion. And the cingulate spinal projection may carry the input that drive the motivation underlying intentional choices to navigate different routes during locomotion. Another interesting module I would like to discuss with you is the ventral premotor spinal module, which binds feature for defensive behavior. And the central nodes of this module are the ventral intraparietal area VIP and the ventral premotor area F4, which are reciprocally connected. The main inputs to VIP come from the vestibular insular parietal cortex and from the vestibular parietal sylvian cortex, as well as from the areas MT and MST from the superior temporal sulcus, which are visual motion sensitive areas. And therefore, these two sources of input properly confer the vestibular and motion sensitivity, visual motion sensitivity to VIP. The input from second somatosensory cortex and from area PF in the inferior parietal lobe confer somatosensory sensitivity to VAP. Area F4 receive a strong input from second somatosensory cortex from area PF and also from a part of area F5. And finally, the cortico-spinal projection, this model, stem from area F4 and consists of small and slow conductive axons. Which are the functional properties that confer to this ventral premotor spinal module a role in defensive behavior? Neurons in area MT, which is a visual motion sensitive area, are sensitive to the direction and speed of visual stimuli approaching body parts, in particular the head and the eyes. MT project to VIP, where neurons have bimodal and match, that is, in register visual and somatosensory receptive fields, since they respond to visual stimuli which move toward the body part where the somatosensory receptive field is located. In other words, neurons in this area show congruent visual and somatosensory direction selectivity. Very similar results are obtained in area F4, where neurons are instead independent from the eye position and therefore from the angle of gaze. The idea underlying all these experiments and the take-home message is that cell with specially aligned somatosensory and visual receptive field in VIP and in F4 are a potential neural substrate for impact prediction and defense. Impact prediction is crucial for interception of objects moving toward the body. In this experiment, Merchant, Battaglia-Mer, and Georgopoulos recorded neural activity in motor cortex and in inferior parietal areas PG or PT in monkeys during interception of stimuli moving in a real and apparent motion condition at different speed. And monkeys use an isometric joystick to move a visual cursor from a central position as to intercept a moving target which was starting his movement at about 180 degrees before. In other terms, the interception movement had to occur at about 6 o'clock, while the visual stimulus started to move at 12 o'clock. And the monkey was rewarded if the angle between the cursor and the visual stimulus was less than 18 degrees. As a control, a no-go task was also presented where all this was repeated, but in absence of any interception movement by the animal, and a center-out task where instead 8 different targets were presented in a classical center-out paradigm at 45 degrees angular interval, and the monkey had to exert a force pulse as to bring the visual cursor to them. And the result of this study showed that a population of motor, here shown in gray, and of parietal cells responded, as you can see here, in the real motion and apparent motion condition at different speed, but were not responsive in the no-go and the center-out task. And therefore, this group of cells might be specific for the interception of real and apparent moving stimuli. And the time-varying neural activity in parietal cortex led in time activity in motor cortex. And while parietal cortex activity was mostly associated with the processing of motion stimuli, in motor cortex, it was mostly related to the force pulse that the animal had to exert to intercept the visual target. However, neural activity in parietal cortex also was observed during the interception movement. And finally, and importantly, neural activity was associated with stimulus angle in the real motion condition, particularly in motor cortex, and with the time to contact, which is a very important parameter for the interception movement in the apparent motion condition. The cortical network, which is engaged by approaching visual stimuli, such as a falling ball, which are accelerated by gravity, has been described in this fMRI experiment in Caminiti's lab about 20 years ago. The regions which are activated are vestibular regions such as the posterior insula, the retrosplenial insula, along with region in the inferior frontal gyrus, intraparietal sulcus, and cingulate region at the border with SMA. Therefore, this network is probably the anatomofunctional substrate for the encoding an internal model of gravity. Another interesting aspect concerns the cortical areas which process visual target accelerated by gravity, since they are also activated by vestibular stimuli, as shown a few years ago in Caminiti's lab by Indovina and his collaborators. In the both medial and lateral aspect of the hemisphere, the major hubs were, in the lateral aspect, the posterior insular cortex, the parietal operculum, the intraparietal area 7IP, which is the putative homologue of area VIP in monkeys, and inferior areas 44 and 45. On the medial aspect of the hemisphere, hubs were 5L and 7A, which are the putative homologues of area PGM in monkey, and as well as area 31 in the retrosplenial cortex, and a number of cingulate areas, in addition to subiculum and entorhinal cortex. The presence of spatial aligned somatosensory and visually receptive fields in areas VIP and F4, considered as a subset for impact prediction and defense, somehow predict what the effect of microstimulation of this area could be. Long-train microstimulation of area VIP results in a constellation of movement, including eye closure, facial grimacing, head withdrawal, elevation of the shoulder, and movement of the arm toward body parts such as face, mouth, eyes, and beside the head or the shoulder. And complex action resembling defensive behavior were also obtained after F4 microstimulation. Lesion studies showed that suppression of VIP through injection of mushimol result in decreased defensive responses, while the activation of VIP after bicuculin injection strengthens defensive reaction. Interestingly, lesion of VIP results in peripersonal neglect. Concerning lesion studies in humans, Body ownership is an important aspect since peripersonal fields and neural activity in VIP and therefore have been somehow linked to the perception of ownership and location of body parts and even of the whole body. And the reduced feeling of ownership over body parts and of defensive responses have been observed after VIP and surrounding white matter lesion. And the damage of this area within this model can also result in somatoparaphrenia, which is a condition characterized by the belief that one's body part belongs to someone else. And this condition is also associated somehow with the impaired defensive behavior. An alternative perspective, but which after all point in the same direction, relies on the value of defensive action. These can be or are considered as an excellent indicator of ownership of a body part, especially from an anthropic perspective. Defensive action are those which oppose very fast, dramatic increase in an animal entropy, and therefore they are aimed at preventing the body from becoming damaged and disorder, in other terms, dismember. Coding of numerosity in area VIP as well as in prefrontal cortex, where the behavioral strategies are supposed to be encoded, is an important function for defensive behavior. Numerical judgment, in fact, is crucial for the type of defensive reaction in front of different number of threats. Should we stay or should we go? Better we leave or better we attack when we are faced with a threat such as the one shown in this slide. The cortico-spinal projection of this model stem from area F4 and conduct information at a rather slow rate, which is 6 meters per second, while the information which is conveyed by motor cortex to the spinal cord goes at about 15 meters per second. Therefore, it is reasonable to believe that the F4 spinal projection can shape the postural reaction which are more suitable for escape and or attack behavior to better set the motor periphery to fast upcoming command from motor cortex when a decision is taken. This is the final part of my presentation, which addresses a rather complex issue concerning the interaction between models and the role of primary motor cortex in model recombination for novel tasks. Hierarchical reinforcement learning suggests that models offer a library of tools that can be dynamically recombined to solve novel tasks. In this experiment, mice that learn two hierarchical subtasks, an object manipulation task and a leaking task, were able to quickly recombine pre-learned action value representation of the neural activity underlying both tasks to learn in a very efficient way the new composite task, as it is shown by the learning curve that they displayed relative to animals who had not been pre-trained in the subtask 1 and 2. Concerning neural activity, calcium imaging traces from individual cells were obtained in different cortical areas such as M1, M2, first somatosensory cortex, retrosplenial and posterior parietal cortex, and the Q action value representation of neurons were computed. It must be said that in deep reinforcement learning model, the so-called Q function estimate the expected reward of taking a particular action in a given state. That is, it considers both the state and the action as input. The Q function is used to express the action value and learn an optimal policy which maximizes the expected cumulative reward. Concerning the results, the cortical distribution of neurons encoding action values in the Q task 1, the object manipulation, and the Q task 2, the leaking task, show a significant increase in the proportion of neuron encoding action value in the transition from the naive to the expert stage of learning. In the composite task, the cortical distribution of movement and leak-related neurons were like those of the in the early stages, were like those of the expert stage of the original subtask. While the transition from the early to the late stage, in other terms, as learning progressed in the composite task, an impressive reorganization of cortical modules occurred from a more dedicated to a more distributed network. In other words, the concerted action of individual neurons in the brain led to a new policy composition and to a model recombination. Another set of studies suggested where the early binding of modules might take place. In this experiment, the forelimb region of M1 was inactivated by photoactivation of parvalbumin-expressive GABAergic inhibitory interneurons after injection of channel rhodopsin 2. And while inactivation impaired forelimb movement during the early and middle stage of motor learning, it had no effect whatsoever in the late stage, and the M1 disengagement from the task correlated in a positive fashion with task performance after learning. In other words, the less M1 was involved in the task, the better the task was performed, suggesting that this is not a simple function of training, but instead of the level of expertise. A similar effect is seen in human. M1 bold activity decreases as fast learning progress in a precision grip task, as does activity in the frontal pole and dorsolateral premotor cortex. In the left primary motor cortex, activity remains almost at a constant level, while in supplementary premotor cortex, it increases slowly and reaching a peak and then it decreases. A marked decrease is observed in the intraparietal sulcus. Therefore, M1 might serve as a major hub that brings parallel models together in the specific context of novel task. And once an animal learns a particular task for long enough, a dedicated network is formed that bypasses M1. This would be reflected in the increased activity of the other motor areas and simultaneous decrease in coupling with M1. And in fact, functional connectivity in humans shows exactly this pattern. Dorsal premotor cortex, supplementary and presupplementary motor cortex are more strongly coupled to M1 during early rather than late sequence learning, as shown in this image. Even sleep-dependent improvements of early learning coincide with decrease in bold M1 activity. And this suggests that the recombinatory pattern, which is learned in motor cortex, is used to consolidate a network that bypasses motor cortex during sleep, thus weakening the M1 representation in this process, which is in keeping with the way hippocampal sequences are used to consolidate memories into the cortex, while at the same time weakening the sequence representation in the hippocampus. Finally, the concept of M1 functioning as an early recombinatory hub also aligns with the variety of conduction velocity and delays of its cortico-descending spinal tract. M1's recombined signal in fact must catch up with those addressed to the spinal cord along other corticospinal tract to ensure the coherent motor command reach the muscles and support the corticospinal modularity hypothesis. I would like to thank Alexandra Battagliamai, Rory Bufacchi, and Giandomenico Iannetti for sharing with me the responsibility of the corticospinal modularity hypothesis. I also like to thank Elena Borra and Giuseppe Luppino for their expert advices and skilled participation in a number of experiments concerning cortical connectivity, and Bruno Haverbeck for providing advanced tool for their quantitative analysis. And a special thanks go to Giorgio Innocenti, who passed away almost 3 years ago. I met Giorgio when I was a third-year medical student at the University of Catania, and he was a young assistant professor. We collaborated for almost 30 years or even more at the early and late stage of my career in Lausanne, Stockholm, and in Rome, finally. What I learned from Giorgio was the importance of looking at neuroanatomy from a physiological perspective and the crucial role that evolution and development play in the understanding of cortical organization and function. Finally, thanks to the audience of this lecture for the attention and patience.